right, so we're going to stay on the theme of the hippocampus and the amygdala. So it's really my pleasure to introduce Dr. Frances Xia, who's a postdoc in my lab. She started in 2019. She got her undergraduate degree at the University of British Columbia and then moved to the University of Toronto, where she did her PhD, an incredibly successful PhD working with Dr. Paul Franklin, identifying kind of the neural circuits and cell types that mediate hippocampus and cortex interactions during consolidation of remote memories. And so she joined. We were very lucky to recruit her to our lab in 2019, where she kind of joined this group, as Andrew mentioned, the Dolby group, to try to back translate some of these findings that you heard about from Josh into animal models. And so she kind of single-handedly in my lab kind of started a whole new technique and a whole new kind of direction, applying kind of high-density electrophysiology techniques in mice, kind of newest techniques out in the field in systems neuroscience, to kind of ask these questions in animal models of depression. And so I think she's got some an incredibly beautiful data set, and so I'm really excited for her to share this work with this community.
Thank you, Mason, for the introduction, and thank you guys for having me here today. And thank you, Josh, for the excellent talk. So now I can just jump right in and share with you some of our work trying to identify and understand biomarkers for mood disorders, trying to understand or decode stress susceptibility in the extended amygdala network. So one of our core research topics is, as I said, trying to better understand the neural circuit mechanisms underlying major depressive disorder. Does this sound weird, or are you guys okay? Okay. So specifically, we're trying to better understand anhedonia, which, as you all know, is the reduced ability to experience pleasure, and it is a core feature of MDD. And existing research has shown that dysfunction in brain circuits underlying reward processing are highly implicated in anhedonia. And specifically, we know that abnormal processing in the mesolimbic dopaminergic system is very important. However, a lot of recent research have also pointed to the potential role of the extra amygdala network. And specifically, we know that the amygdala is anatomically very well connected with regions such as the ventral hippocampus, medial prefrontal cortex, nucleus accumbens, and thalamus. These regions, as well as connections between them, are shown to be very important in processing both aversive and appetitive information, and that activity in this network can really modulate anxiety and mood-related behavior. However, the role of amygdala and its connected regions in anhedonia remains unclear. So here we would like to address two main questions. One is, how does stress modulate the representations of reward outcomes in the extended amygdala network and affect reward-seeking behavior? And secondly, can we rescue physiological and behavioral signatures of anhedonia via circuit-specific manipulations? We explore these questions in mice using a chronic social diffuse stress paradigm, or CSDS. This is a paradigm that is very well established in rodents to induce depression-like behavior, and specifically different stress phenotypes. So in this task, a mouse is defeated daily by a more aggressive strain of mice for 10 days. And then on day 11, we run a social interaction test to assess social avoidance. And then after that, we have fixed the mice and asked them to perform a sucrose preference test aimed to assess anhedonia. During this task, we perform neuropixels recordings, which is a technique that is very similar to a high-density silicon probe that allows us to record simultaneously many different neurons and across different regions. So the task structure is very simple. The mouse is water deprived, and it's head fixed and has two lick spouts in front of it, one delivering water, one delivering sucrose. The mouse can freely choose which reward it wants to receive at any given moment. The only limitation is that after it receives one reward, then there's an intertrial interval of 8 seconds. So it needs to wait for 8 seconds before the next reward becomes available. At the end of 8 seconds, if the mouse licks, then it will receive that reward. So in other words, the rewards are lick contingent. If the mouse does not lick, at the end of 8 seconds, it will not receive any reward. In total, we perform 150 trials per session per day, and we repeat across multiple days, counterbalancing the spouts. In total, we collected data from 67 mice, in both control mice that are non-stressed and also in stressed mice. And we have over 24,000 neurons across 48 different brain regions, as you can see here. But today, I will be mainly focusing on our work in trying to understand better the neural signatures in amygdala and ventral hippocampus. First, I'll show you the behavior results. So here we have two main behavior metrics. One is sucrose preference, that's on the y-axis, and this is basically the amount of sucrose rewards the mice receive out of total number of rewards. And on the x-axis is our second behavioral measure, that's the social interaction ratio, where the higher the ratio, the more social interaction the mouse exhibits. And you can see that these two behavior measures are strongly correlated in the stress group shown in green. And we did this experiment using both male and female mice, and you can see very similar patterns in both sexes. However, you can also see that there's a huge variability in the behavior here in the stress group. So we'd like to better understand the variability in this behavior. So we perform unsupervised k-means clustering, trying to pull out unique clusters. And here we identify two clusters. One is the susceptible cluster in blue. They have lower sucrose preference and lower social interaction ratio, and then the resilient mice on the upper right quadrant. We looked at another behavior metric, and we see that susceptible and resilient mice are very distinct. So what I'm showing you here is the lick rate discrimination index, where the higher the DI, the more the mice lick for sucrose trials than they do for water. And you can see that's the case for resilient and control mice, but not so much for susceptible. We also looked at single neuron firing differences in different regions across groups, and we found that the differences are region specific. So for example, in BLA or basolateral amygdala, here both stress groups, so susceptible and resilient, shows suppressed firing in comparison to controls. In contrast, in ventral CA1, the susceptible group uniquely shows elevated firing. The second metric we explored at the single neuron level is reward choice selectivity. And that's because we noticed that there are cells like this one, cell 1 on the left, that shows very similar firing rate profile for both sucrose and water trials. And we also have cells like the cell 2 here that show preferential firing for one of the trial types. So to quantify this reward choice selectivity, we calculated the area under the receiver operating characteristic curve, or AURC. Very simply, if AURC is greater than 0.5, that means the cell is trial type selective. And we do this calculation for every single cell and then look at the distribution of AURC and compare that between groups. We focused our analysis on this baseline period, which is -4 to -3 seconds before reward delivery, and also this 1 second post-reward period. What we found was that very surprisingly, even though the susceptible mice showed reduced sucrose preference, in BLA, they actually showed increased AURC or increased reward choice selectivity. And we also observed similar pattern in the resilient mice. We saw this during both baseline and post-reward period in BLA, and very similar pattern in ventral CA1. So here we found this very surprising thing that the stress mice actually show increased single neuron reward choice selectivity in BLA and ventral CA1. So next, we wanted to explore this further at a population level. So we decided to run a population decoder for trial types. So first, let me tell you how we define a trial type. As the mouse is going through this trial, it can freely choose what reward it wants at time 0. So if it chooses sucrose reward at time 0, then we will retrospectively define -4 to 4 seconds around that choice as a sucrose trial, and similarly for water. And the reason why we did this is because we want to see if we can use the neural activity data to predict what the mouse will choose even before they physically make that choice. So we take the firing rate of all the cells for water trials and also for sucrose trials, and then we train a linear support vector machine classifier trying to distinguish the population representations for sucrose trials and water trials. We train the decoder using data from one time bin and test it on held out data within the same time bin. One prediction is that maybe at the neural level in the stress mice, especially the susceptible mice, maybe the representations for water and sucrose are intermingled. This will lead to at-chance decoding accuracy at 0.5, and this lack of neural discrimination is maybe what's leading to this reduced sucrose preference. Alternatively, maybe at the neural level, the representations for water and sucrose are very distinct. This will lead to a high decoding accuracy, but maybe this is somehow actually maladaptive for behavior. What we see with our data is actually more similar to the second case. So what I'm showing you here is the decoding accuracy in BLA for the control mice. you can see that 4 seconds before reward delivery, the decoding is at chance, then it gradually increases and peaks after reward delivery. In contrast, in the susceptible group, the decoding accuracy is already very high at its baseline period and just remains stable throughout the entire course of the trial. So the way that you can get a very high decoding accuracy at baseline suggests that the susceptible mice, at least the BLA neurons, already know 4 seconds before the choice that which choice the mouse is going to make. And the resilient mice lie somewhere in between. So you can see that they share some characteristic with the susceptible. They have this very high baseline decoding, again, suggesting that they already know which choice they're going to make, but the decoding still increases post-reward. So to summarize, 2 main observations here. One is that we have this very high baseline decoding. To us, this suggests an inflexibility in the representation of future reward because these stress mice already know which reward they're going to choose, even 4 seconds before they physically make that choice. And secondly, we saw in susceptible mice that the change in decoding accuracy from baseline to post-reward is very small. So to us, this suggests a reduced sensitivity to the current reward. We saw this pattern in BLA as well as in ventral CA1, as you can see here. And this is quantified on the right. We see this very high baseline decoding, again, suggesting inflexibility in representation of future reward. But this in susceptible group is accompanied by reduced change in decoding accuracy from pre to post, suggesting reduced sensitivity to current reward. So far, I focused on activity within each region, but we're also very interested in the interaction between ventral hippocampus and amygdala. So what we did next is we calculated the region average firing rate in each region and then correlated the activity across regions. We did the correlation for sucrose trial and separately for water trial, and then calculated delta correlation or changing correlation. So what this changing correlation tells us is the degree of interaction between these regions in sucrose trials in contrast to water. And what we found is that the degree of this interaction, between amygdala and ventral hippocampus is positively correlated with sucrose preference, suggesting that in mice that have greater correlation between these two regions in sucrose trials, they are also the mice that have greater sucrose preference. So this led us to wonder, what if we can somehow boost the interaction between these two regions in these mice in the lower left quadrant? Can we then somehow rescue their sucrose preference? So we did this via a chemogenetic approach, which allows us to specifically transfect the projection neurons from ventral hippocampus to amygdala with a synthetic receptor. Then we can later go in to activate this receptor at a temporally specific manner. So we did this exact same experiment, but now during the recording period, we have 2 phases. During phase 1, mice receive saline, which is a control injection. Then we give the CNO, which is the synthetic receptor actuator, and then run the 75 trials. So the second period is when we're able to activate the ventral hippocampal neurons that project to amygdala. And what we found is that CNO, or activation of this pathway, was able to increase the interaction between amygdala and ventral hippocampus. We're also able to increase the decoding accuracy for reward types in both BLA and ventral CA1. And importantly, this was able to increase sucrose preference in these mice. So this shows that activation of the ventral hippocampal-amygdala pathway was able to rescue physiological and behavioral signatures of anhedonia. So to briefly summarize this part of the talk, I showed you that at a population level, in the susceptible mice, we see inflexibility in the representation of future reward outcomes and reduced sensitivity to current reward outcomes in both amygdala and ventral hippocampus. And I also showed you that activation of the ventral hippocampal-amygdala pathway can rescue signatures of anhedonia. And then in the last few minutes, I'll share with you some of our ongoing work where we're trying to see if we can identify susceptible mice using neuroactivity patterns, even in the absence of task. So all the data I showed you is from this task period where the mice have to do this sucrose preference test. But during the recording, we also have a 10-minute pre-task period where mice are head fixed, but they're not doing any behavior. And the goal here is to see if we can pull out neural signatures during this behavior-free period to already tell us whether the mouse is susceptible, resilient, or control. And specifically, what we want to analyze is the geometry of population representations. And what I mean by geometry is that imagine you have 3 neurons, F1, F2, and F3. If you plot their firing rate in this 3D plot, their firing rate at time t can be represented by this single dot. Then at t+1, you have this other dot, and then t+2, we may move up here. So now you have a triangle. This triangle is then a geometric representation of the population activity of these 3 neurons at these 3 time points. But of course, if you have, say, 100 neurons and 50 time points, this will be a 100-dimensional space with a geometric object that has 50 anchor points. But that's too many dimensions to visualize. So here, what I'm showing you is the first 3 dimension of one example mouse and its activity during this pre-task behavior-free period. And you can see it forms a very unique geometric object. And we can do this for every single mouse. But the question comes down to how do we effectively compare these geometric objects across mice? And one approach that we did is principal component analysis, or PCA, which helps us to identify the embedding dimensionality, or how high dimension the population data is. Using PCA, we're able to pull out this measure called participation ratio. And a high participation ratio is indicative of a high dimensional population representation. And what we found is that in BLA, the susceptible group have higher participation ratio, which means that they have higher dimensional population representation in comparison to controls. And resilient mice lie somewhere in between. In contrast, in ventral CA1, we actually do not observe any group differences. The second measure we looked at is something that tried to quantify population activity states. And we did this using a hidden Markov model. The assumption for this analysis is that the observed or the reported data are the results of underlying hidden states. And HMM allows you to pull out these hidden states and look at the transition between them. And here is an example control mouse and its hidden states in BLA. You can see the hidden states on X and Y axis. And in addition, I'm showing you the correlation between these states. You can see that even though there are a number of states we identified, a lot of these states are very similar to each other. They have very high correlation. In contrast, in this example, susceptible mouse, all these states seem much more distinct. We can quantify the number of unique states by setting different thresholds. So here I'm showing you a clustering technique where on the y-axis, I'm showing you 1 minus the Pearson correlation coefficient. So at 0, we actually have the highest correlation threshold. At this threshold, the state is only correlated with itself. But as you keep moving up the y-axis, then more similar states get clustered together. If we set, say, threshold 0.2, in this control mouse, we can identify 4 states. But in the susceptible mouse, we can identify 13 states. So this example shows you that, at least in this example mouse, susceptible mouse has greater number of unique hidden states than the controlled. And this turns out to be true across all the mice that we looked at. So this is the group data. I'm showing you the percentage of clusters retained across a variety of thresholds. You can see that susceptible mice have greater number of these unique hidden states in comparison to controls, with resilience somewhere in between. We can also look at the duration of each of these states. You can see that the susceptible group have the shortest state duration. So in other words, the BLA activity in the susceptible mice not only show greater number of these hidden unique states, but they also have higher transition frequency. And the goal for identifying all of these features is to see if we can use these features to then identify whether a mouse is susceptible, resilient, or controls. And the answer is yes, indeed, we can do that using BLA data. So here we build a decoder using these features listed here: firing rate, mean, and standard deviation, the first 3 principal components of the PCA analysis, and the first 5 clusters in HMM. When we use all these features and trying to identify control versus susceptible mouse, we can do that very nicely with almost 90% decoding accuracy. And then we systematically removed each of these features to see how much the decoding drops. And the idea is that if we remove one feature and the decoding now drops to chance, that means that one feature alone is responsible for the high decoding. So for example, if we remove firing mean, the decoding drops to 80%. So this feature is fairly important. And similarly, the standard deviation and the first 3 clusters of HMM. We did a similar analysis trying to decode other pairs. But it turns out that we can just barely decode above chance for control versus resilient, and not at all for susceptible versus resilient. We also did similar analysis in ventral CA1, and interestingly, we actually cannot decode. So this suggests that these 3 features that we used here are uniquely important to trying to identify different groups in BLA. And the second thing this suggests is that since we can decode control versus susceptible very well, it suggests that maybe in this feature space, these two groups are the most distinct, but resilient may be somewhere in between. So we visualize this using multidimensional scaling, or MDS. It's another method of dimensionality reduction. And here you can see that in this feature space, indeed, the control and susceptible are the most distinct from each other, with the resilient right in between, except this one outlier. So what I showed you in this last section is that, yes, indeed, we can identify susceptible mice using neural activity patterns, even in the absence of task. And specifically, we can do this in amygdala. So finally, I'd like to thank all the key players in this project: Nina, a technician in our lab, Mason, my mentor, and Valeria and Stefano, our collaborators in Columbia University, as well as our funding sources. And thank you, everyone, for having me here today.
Wonderful. Wonderful talk. Thank you very much. Actually, both talks were fantastic and really interesting. May pose a question here that is not answerable at this moment, but just thinking about the two talks together. So in the first talk, we heard about the notion that this sort of underlying biomarker reflection of circuit level activity between hippocampus and amygdala leads to this wide range of subjective experiences in humans. And then in the second talk, what we heard about is that you could start with a task, generate a behavioral output that's related to a circuit, and then start to really understand sort of what the component parts of that are. That, by the way, I would just say alone is like the fulfillment of a dream for many of us who are the older generation sitting in this room right now. But the question I have is around the notion of kind of your conceptualization of what you're doing is looking at anhedonia. Because from the human data, what you would, I think, anticipate is that you're pushing the system, leading to changes in circuit that are leading to a measurable behavioral outcome. But when you bring in the word anhedonia, what you're bringing in is sort of reflecting this subjective experience among humans with depression that we just learned in the first talk, is not necessarily sort of a one-to-one relationship with that circuit level activity. So I guess what I'm wondering is, what's the value of thinking about this as anhedonia, as opposed to we're altering, reward-based behavior in mice, and then we're going to try to understand what those subjective states are as we translate to humans. Does that question make sense?
That's a very good question. I mean, at the end of the day, what we are assessing is reward-seeking behavior. However, these mice, as we put them through this CSDS paradigm, they don't just exhibit this type of reduced reward seeking, for example, in the susceptible mice, but these mice also exhibit other depression-like symptoms. For example, they show high anxiety, and we also see reduced social interaction. So here I'm only showing you the reward aspect,
yes,
but they do exhibit many other characteristics that are shared in depression-like behavior, or as much as we can call it in rodents.
Yeah. So, I mean, I think it's just a really interesting opportunity for us, potentially. I mean, honestly, you get this into Cell and Nature by giving some insight into anhedonia. I think, the editors are going to ask for that. But I think in the discussion, I mean, in all of our discussions and thinking about this back translation, I think we really have an extraordinary opportunity to start thinking more deeply about anthropomorphizing and about actually being able to go back and forth between this incredibly detailed understanding at the circuit level and a much more detailed understanding now, because of these intracranial recordings in humans, about the kind of the broad range of symptomatic manifestations that might lead to.
Yes, definitely. And right now we also have other work in the lab that are trying to use more complicated behavior tasks that may be a bit more similar to what we do in human patients.
Incredibly exciting. Thank you.
Great talk. Lots to think about and ask about. I'll just drill on one thing that's related to what Matt was talking about. As I understand it, Vikas has sort of done the opposite thing. He's decoupled amygdala and hippocampus in a different task. This is the elevated plus maze exploring task, and found sort of therapeutic, if you will, effects of that, if I'm not mistaken. Is that correct? Out of phase. What he did is he drove them out of phase. Was that right?
VCAS desynced GABAergic interneurons in VLA and in ventral hippocampus and basolateral amygdala, and showed that it reduced some anxiety behaviors. Yes,
but this points out that it may be neuron dependent or task dependent, or I mean,
or both.
so just thoughts.
Yeah, so I think it really depends on the specific neurons, and then also it's a different measure. So here we didn't specifically look at the beta coherence between amygdala and ventral hippocampus, so that could be one thing. And also, I guess just the way that VCAS decoupled may be different.
So this just makes me reflect on the human findings and the question of stimulating these regions very broadly. We're not doing specific circuit or specific cell type stimulation. And this may account for heterogeneity in what we're getting because our electrodes are in different places in different people. So before concluding that a single biomarker is linked to a different set of symptoms, we need to figure out if we're getting actually different biomarkers in different people because our electrodes are in slightly different places.
or even just recording a greater epiphenomenon that, there's lots of different cell types may together produce, beta coherence. And so what are the specific, maybe there's different cell types that are contributing to it, or in different microcircuits in each patient, which is why, Francis, your work is so awesome.
That this kind of goes back to the question that I asked Josh during his about the on task versus off task, because I think that some of the cool stuff that Francis is finding now is that we get actually in the amygdala very different signatures when the animal's not doing anything versus on a task. And you mentioned VCAS's work. A lot of that coherence measurements were done off task, like when the animal's off task, right? And so I think that there's some really interesting things there that that should be explored.
When you show, this is a question about the decoding.
Mhm.
And so you show kind of like the time period before the task and after the task. And so does that mean that what that is that a measure of activity? Like activity in that specific neuron is not predictive until a certain time point, and at that time point, activity in that neuron becomes predictive?
It's more at the population level.
Okay.
So over there, we're taking all the neurons and then their activity in water trials and their activity in sucrose trials. And we see that the representations at the population level are very distinct.
Mm-hmm.
So this could be traced back to when I showed the single neuron selectivity measure, where we also see this enhanced single neuron reward choice selectivity during the same window. So that could contribute to decoding. But we actually did try multiple methods trying to nail down what drives the high decoding, and it turns out to be a very difficult task.
So unfortunately, we're out of time, but I want to congratulate our two awardees and speakers, and great talks to both of you.